Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic control and coordination. So in the previous lecture, I've given you a brief idea about what is coordination, how the coordination is done in the living organisms. So coordination involves, uh, you can say, the working together of all the organs as well as the organ system. Okay, so because of this coordination, the vital processes of the living organisms are carried out. Then apart from this, we also studied about the movement in the plants. Okay, so plants, they are still, but the parts of the plant can move. So we studied about the phototropism, geotropism, as well as thigmotropism, and we also studied about the chemotropism, where the stimulus is chemicals. Now stimulus, there were two kinds of stimulus. First one, the external stimulus. Another one was the internal stimulus. And in the end, we studied about the phenomena of photoperiodism. So I've given you a brief idea about photoperiodism, and I've also given you idea about the short day plants, long day plants, as well as the day neutral plants. So in this lecture, we'll be studying about this short day, long day, and day neutral plants in detail. We will see how we can define them. We will see the examples of these kind of plants. Okay. And then later on, we'll study about the phytohormones, also known as the plant hormones. Okay. So let's start with this lecture. So in the previous lecture, I've given you a brief idea about the photoperiodism and how it is related to the flowering. Okay. So one of the response controlled by the photoperiodism is the flowering, and some other responses are seed dormancy, or you can say germination of the seeds, and plant growth. So these are all dependent on the relative day, okay, or the relative length of the day as well as the night. All right, this is clear. This is night. Now let's again understand photoperiodism. So it is a physiological change occurring in the plant in response to relative length of day and night. And what was photoperiod? Photoperiod is the relative length of day and night, and any physiological change occurring in the plant, okay, due to this relative length of day and night. So relative length of day and night, this is known as photoperiod. Okay, and the physiological change, or you can say the biological response. Okay, physiological change means the biological response. Biological response can be induction of the flowering or the germination of the seeds. All right. So this was known as the photoperiodism. The photoperiodism term were used by Garner as well as Allard. Garner and Allard. Okay. This is clear. Now, on the basis of the photoperiod, there are three classes of plants. One is the short day plants, long day plants, another category, and third is the day neutral plants. Now we'll study about these kind of plants individually. So let's start with the short day plants. What are short day plants? Now here in this short day plants, flowering occurs when the duration of the daylight, okay, or you can say the photo period, okay, it falls below a specific critical threshold. Okay, so this critical threshold is also known as the critical photo period. Okay, you can also call it as critical photo period. Okay, so these short day plants, they flower when the duration of the day, you can say, or the duration of the daylight, it is less, or it falls below this critical threshold, this critical photo period. Okay, this is clear. So suppose this is the length of the day. Okay, a day is of 24 hours. So this is the critical photo period. Okay, here. So this represents the critical threshold or the critical photo period. All right, then these plants will flower when when the day length is less than the critical photo period. So this should be the day length here. Okay, it is less than the critical photo period. So the day or the daylight or the day length is here less than the critical photo period. Okay, if the day length is above this critical photo period, then it will not flower. All right, this is clear. Examples are chrysanthemum, rice, soya bean, and onion. Clear? Now let's talk about the long day plants. So they are just the opposite of the short day plants. Here, the flowering will be induced when the duration of the day or the duration of the daylight, it exceeds the critical threshold or the critical photo period. So again, if this is the day length, which is of 24 hours, then if this is the critical photo period, if this is the critical threshold or critical photo period, then what happens? If the day length is more than this critical photo period, so if this is the day length, this one, okay, the day length here is more than the critical photo period, then in this condition, it will flower. Okay, in this condition, it will flower because the day length is more than the critical photo period. Okay, but if the day length is less than this critical photo period, then it will not flower. Clear? Examples are lettuce, spinach, radish, sugar beet, and potato. Okay, so here you can write it will flower when the daylight or the day length is more than the critical photo period. Clear? Okay, so day length is more. So suppose in 24 hours, you have 14 hours of day and 10 hours of the night. Clear? Now let's talk about the day neutral plants. So these day neutral plants, they are not affected by the relative length of the day as well as the night. So these plants, they undergo a flowering process which are unaffected by the day length. Okay, so how do they flower? They flower at a certain age. Whenever they reach a certain age, then they will be automatically flower. Okay, so instead they bloom solely when they reach a certain age. Okay, so these plants are called day neutral plants because they are not affected by the photo period, or you can say they are not affected by the length of the day or the night. What are the examples? Examples are cucumber, corn, tomato, pea, as well as dandelion. Clear? Now, this is an image showing you the flowering in response to the photo period. Okay, so here you can see in long day plants, what is happening here? This is the critical photo period. So long day plants, they will flower when the day length is more than this critical photo period. So when this day length, you can see above, if the day length is more than the critical photo period, then it will flower. And when the day length is less than the critical photo period, or it is below the photo period, then it will not flower. Then short day plants, this is the critical photo period. So they will flower when the day length is less than or below the critical photo period. So here they will flower. Whereas if the day length is more than or above the critical photo period, then they will not flower. Okay. And the day neutral plants, they are independent or they are not affected by the relative length of the day. Okay. So they will flower. Okay. Whenever they reach a certain age. So they will flower after vegetative maturity. So, there are two phases in the life cycle of the plant. Okay, so what are the phases? First phase is the vegetative phase, in which the growth of the plant takes place. And second is what? Second is the reproductive phase. So whenever these day neutral plants, they when they attain the vegetative maturity, then automatically they will flower and they will enter the reproductive phase. Okay, this is clear. Now let's move on to the next topic, which is vernalization. So vernalization is also related to the flowering, or vernalization can also induce flowering. Clear? Now the term vernalization, it was coined by Lysenko for promotion of the flowering by a cold treatment. So here what is happening, you give the cold treatment to the plant, and because of this cold treatment there is induction of the flower. Okay, or there is induction of the flowering. So induction of the flowering by cold treatment, this is known as vernalization. Okay, and what is the most appropriate temperature or suitable temperature for vernalization? It is 4 degrees Celsius. The plants are exposed to 4 degrees Celsius for a time period of 4 to 3 months. Okay, now for flowering in the winter varieties, so winter varieties, they grow in less temperature or low temperature. So what happens? How we can induce flowering? So we can give them cold treatment. So cold treatment is necessary for flowering in the winter varieties because they grow in the less or the low temperature. Okay, now in nature, plant requiring the cold treatment, they usually behave as biennial. So this you have to remember. The plants that are biennial, okay, they require cold treatment to enter into the reproductive phase. So I've told you one was the vegetative phase, in which the growth of the plant takes place. So there is a growth of stem, the branches, the leaves, that takes place during the vegetative phase. And during the reproductive phase, there is formation of the flower, fruits, seeds, etc. So in biennial plants, or okay, what happens? Cold treatment is required to transform from vegetative to the reproductive phase, or you can say the induction of the flower requires the cold treatment. Now, these biennial plants, they germinate and grow vegetatively in the first season, and they produce flowers during the second season after getting the cold treatment. So they require cold treatment for inducing of the flower or for the transformation from vegetative to the reproductive phase. Suitable temperature, I've already told you, it is 4 degrees Celsius, and the time duration is also important here. For how much duration you have given this cold treatment. Both of these factors matter. Now let's have a look at the difference between phototropism and the photoperiodism. So both of the terms we have discussed in the previous lecture. Now let's have a look at the differences between these two. So both are related to the light because both contains the photo. So photo means light. Now tropism means movement, we have seen. So it is the tropic movement, or you can say movement towards the light. So movement in response to the light, this is known as phototropism. Here the stimulus is light. So light acts what? Light acts as a stimulus. Okay. Now what is photoperiodism? It is the biological response or the physiological response to relative lengths of day and night. Clear? This is photoperiodism. Now the stimulus, which is the light, it is perceived by the apical meristem. Okay. And here the stimulus, it is perceived by the leaves. Okay, this is also clear. So during photoperiodism, the apical meristem, what are apical meristem? Such as shoot apical meristem, this perceives the stimulus, which is the light. Here stimulus, again here light is the stimulus, which is perceived by the leaves. Okay, and we have discussed which pigment is involved here. Phytochrome is involved here, which exists in two forms, PFR as well as the PR. Clear? Now, phototropism, it is due to the differential growth in the elongation zone. So what happens? There is a curvature growth. We have talked about this term, curvature movement, okay, or curvature. So movement due to the formation of this curvature is known as curvature movement. So what happens? There is a differential growth. All right. So what happens due to the uh, due to the this is due to the hormones such as auxins. So what happens? These hormones such as auxins, they accumulate on one side. Okay, like if the auxins are produced in the shoot tips, so they accumulate on one side, and wherever they accumulate, okay, then there is a cell division there. And what happens due to this? And on the other side there is no cell division. So on the side where rapid cell division is taking place due to the presence of the hormone, that side will bend a little bit, it will show curvature. Okay, so if initially this is the shoot apical meristem, okay, so this region is the shoot apical meristem, then what will happen in next stage? So due to the differential growth, it will form a curvature. So this is known as the curvature movement, and this is shown during the phototropism. So in this region there will be more growth, or you can say more growth, you can say, and this is all regulated by the hormones such as auxins. Okay, here is more growth. Why there is more growth? Because there is more cell division, as well as there is more cell elongation also, and this is all caused by the hormones such as auxin. So here there is more growth in this region, and here in this region there is less growth. So because of this, the apical meristem, or you can say the stem or the shoot, it will bend, or it will show the curvature. So this is known as the curvature movement, and it is due to the differential growth. So here you can see what is differential growth. In one region there is more growth, in other region there is less growth. Clear? Now photoperiodism, it is due to the replacement of the vegetative buds by the reproductive buds. So I have already told you there are two phases, vegetative and the reproductive phase. So vegetative buds, which give rise to the stem and branches, they are replaced by the reproductive buds, which will give rise to the flowers. Clear? Now let's move on. Now let's talk about the growth regulators. Okay, we were talking about the growth regulators such as auxin, I have told you in the previous slide. Now, what are these growth regulators? These are very important chemicals which affects the growth. Okay, now these growth hormones, also known as the phytohormones or plant hormones, these are the natural growth substances which are produced in any part of the plant. Okay, any part of the plant means they can produce in the shoot apical meristem, they can be produced in the root apical meristem. Okay, and they are then transferred to another part and they influence the growth of the plant. So first is the site of production. So they are produced at specific sites such as shoot apical meristem or root apical meristem, and when they are produced there, then they are transferred to another region and they show their effect in the transferred region. Okay, now the growth regulators. So growth hormones are also known as the growth regulators. They consist of auxins, gibberellins, cytokinin, ethylene, and abscisic acid. So there are 5 types of, you can say, growth hormones or phytohormones or growth regulators. Why growth regulator? Because they are regulating the growth. So first one is the auxin, second one is the gibberellins, third is the cytokinin, fourth is ethylene. What is ethylene? Ethylene is a gaseous hormone. Okay, this is the gaseous hormone. And last one is the abscisic acid. Now, auxins, gibberellins, and the cytokinins, these 3 hormones, they are the growth promoters, means they promote the growth. Whereas ethylene as well as the abscisic acid, they are known as growth inhibitors. Clear? Now we'll talk about all these hormones individually. So first, I will give you a brief idea about the growth promoters now. So in this lecture, we'll be talking about the growth promoters and the discovery of the growth promoters. And then in the next session, we'll be discussing about the functions of these hormones, all right, and the other topics as well. Now let's talk about the growth promoters first. So growth promoters are auxin, cytokinin, and gibberellins. So let's talk about the auxins first. So auxins are the growth hormones which were first discovered by Charles Darwin. Now, auxins are the weak organic acid which can promote elongation as well as the growth. Now, these auxins, they are produced in the apical meristem, means the shoot apical meristem as well as the root apical meristem. The tip of the shoots, or you can say tip of the shoot or the stem. So tip of the root is known as the root apical meristem. So these meristem, they are involved in the synthesis of hormone known as auxin. So this is root apical meristem, and in the shoot or the stem we will have the shoot apical meristem. Okay, this is clear. Apart from this, auxins are also produced in the young leaves, flower buds, as well as the fruits. Now, the first discovered plant hormone was identified as indole acetic acid. So, indole acetic acid was the first discovered plant hormone. It is an auxin. And what kind of auxin? It is a natural auxin. So, auxins can be natural as well as synthetic. So, indole acetic acid is the example of natural auxin. There is one more auxin known as the indole butyric acid. This is also a natural auxin. Indole butyric acid known as IBA. Okay, this is also a natural auxin. Then there are synthetic auxin also. Okay, some are synthetic auxin, such as 2,4-D. So 2,4-D, it is a synthetic auxin. What is the full form of 2,4-D? 2,4-dichlorophenoxyacetic acid. This is 2,4-D. It is 2,4-dichlorophenoxyacetic acid. Okay, it is used as a herbicide or the weedicide. 2,4-D, it is sprayed in the agriculture field to kill the weeds or the herbs. Clear? Now, the actual discovery or the experiment that was done to isolate the auxins, that is known as the Avena coleoptile experiment. Okay, it is known as the Avena coleoptile experiment. So here, the Avena plant was used to study the isolation of the auxins. So in the tips of the Avena coleoptile, auxins was being produced. So in the presence of, you can say, light, what happened? This tip Avena coleoptile, there was a curvature. So earlier I have told you, if this is the Avena coleoptile. So whenever it was exposed to the light, it developed a curvature. Okay, so whenever it was exposed to the light, okay, you can, or the sunlight, you can say, then it developed a, so this was the shoot apical meristem. This is the, you can say, SAM. SAM is shoot apical meristem, and there was a curvature seen. So this curvature was due to the presence of auxin, and this developed due to the differential growth. So what they did, they cut this shoot apical meristem and they placed it on the agar block, and then this auxin is diffused into the agar, and then they kept this agar on a another coleoptile in which the shoot apical meristem was removed. And then what happened when they kept this agar block on that shoot apical meristem in which this shoot apical meristem was removed? Then what happened? There was again development of the curvature. That's why, and it was because of the presence of the auxins. Okay, so what was done? The shoot apical meristem region was cut. Okay, this was cut. It was isolated from this coleoptile, and then it was kept on a agar block. So this is the agar block. All right, this was the agar block. This is the shoot apical meristem that was cut. Then what happened? Something diffused into this agar, and this was auxin. And then this agar block was kept on the coleoptile in which the shoot apical meristem was removed. Again, you can draw shoot apical meristem here. Again, you can draw the coleoptile. So this is the coleoptile in which the shoot apical meristem is being removed, and on this agar block was kept. Okay, now this agar block consists of the hormone auxin, and it led to the development of the curvature again here. Okay, again. So this is how auxin was isolated. Now let's talk about the next hormone, gibberellins. Gibberellins, they have a unique property of increasing the height of the plant, but they do not cause the curvature. So whenever you apply gibberellins exogenously, it can increase the height of the plant. How it can increase the height of the plant? By increasing the length of the stem. So it increases the length of the stem. It helps in the stem elongation, you can say. Okay, you can say it helps in the stem elongation. And that's why the length of the plant also increases. Now, the gibberellins were first discovered in Japan by Yabuta and Sumuki. Now, let's talk about the discovery of the gibberellins, how gibberellins were discovered. So, in 1926, gibberellic acid, it was first initially identified by Kurosawa. So, he was doing research on extract from the rice plant, okay, affected by the fungus Gibberella fugicori. Okay, so these rice plants, they were infected by the fungus Gibberella fugicori. So what was happening? So these rice plants were exceptionally very tall. Okay, so the height of the plant was these plants were quite tall, and they were given the name foolish plant. So these rice plants, they were termed as foolish plant because they exhibit considerable height due to the infection. So due to the infection of this fungus Gibberella fugicori, these rice plants were exceptionally tall, and they were given the name foolish plant, and the disease is also known as foolish seedling plant disease. And this was because of the infection caused by this fungus Gibberella fugicori. So Gibberella fugicori was releasing some chemical substance that increased the height of the plant. Clear? So the extract from the infected rice plant, it was applied to a healthy plant. Okay, so the application of the extract from the fungus also, okay, and the plant that were infected with the fungus, so they will obviously contain the extract of the fungus. So the application of the extract from the fungus to another healthy plant led to a significant increase in the height of the plant. Now subsequently, in 1935 and 1938, three Japanese researchers, Yabuta, Sumuki, and Hayashi, they isolated the chemical from this extract, and they designated it as gibberellic acid, which is also abbreviated as GA. Clear? So there are a lot of types of gibberellic acid 1, gibberellic acid 2, gibberellic acid 3, and so on. Okay, so this was the story of discovery of the gibberellins. So you can say gibberellins, it were extracted from the fungus Gibberella fugicori. clear, which infected the rice plant. And the infected rice plant, they show exceptionally, they were exceptionally tall. And why? Because why they were tall? Because of the infection of the fungus Gibberella fujicori, which was releasing a chemical which we now know it as gibberellic acid. Now let's talk about the cytokinin. So these cytokinin, they are chemically basic growth hormones which promote the cell division. So what is the function? What is the main function of the cytokinin? They promote the cell division. Now this group of phytohormones, they were discovered by Miller, and there was another scientist, Cook. So Miller and Cook, they discovered the hormone kinetin. Now cytokinin, or you can say the cytokinin hormone, they were discovered when Carolus Miller, he isolated the crystalline substance from the degraded DNA material. Okay, so this first cytokinin was discovered from the herring sperm DNA. Okay, it was isolated from herring sperm DNA. And Miller was the person who isolated the crystalline substances from this herring sperm DNA. Okay, and this crystalline substance was named as kinetin. Clear? So this is how cytokinin were discovered. Clear? So we have seen the discovery of 3 hormones, auxins, gibberellins, as well as cytokinins. Okay, so all of these 3 are the growth promoters. Now in the next lecture, we'll talk about the discovery of the 2 other hormones, abscisic acid as well as the ethylene. So both of these hormones are growth inhibitors, and we'll also talk about the function of these hormones. Okay, and then we'll talk about the chemical coordination in animals as well. And then later on, we'll talk about the endocrine glands that are present in the human body. Clear? So this was the end of this session. Thank you very much for listening.